So moving on to the next session, delighted to welcome Audrey from the Children's Hospital of Philadelphia. The talk title is Breath-Based Diagnosis of Pediatric Medicine. So over to you, Audrey, and thank you for taking the time. Are you able to share slides? Okay.
Oh, working on it now. Let's see.
Excellent, excellent.
All right, that look okay?
Yeah, that looks great. Over to you, Audrey.
All right. Awesome. Thank you so much, Billy, for the chance to talk to everyone about our work on pediatric malaria. To introduce myself, I'm Audrey John. I'm Chief of Infectious Diseases at the Children's Hospital of Philadelphia, obviously based in Pennsylvania. So happy Election Day again to those who celebrate. I can actually hear the DJ from the local polling place from where I am. So it's an exciting day for us, but also pleased to get a chance to talk to you about some of our fun science. And as Andy Cow introduced already, we've been long interested in breath-based diagnostics for pediatric conditions. Andy did a phenomenal job talking about our work on the microbiome, and I'm going to talk to you about our work on pediatric malaria. So to introduce this disease to those of you who may not be familiar, malaria remains an incredible global health problem. So there are roughly 225 to 250 million cases of malaria each year. And over 500,000 deaths still every year around the globe to this disease. And it's really a disease that disproportionately affects children. So about 3/4 of the malaria deaths are due to children, are in children under the age of 5 or in pregnant women. I'm a pediatric infectious disease physician, and I've long been impressed by the contribution of infectious diseases to childhood mortality. You can see that graph on the left from UNICEF showing the incredible proportion of infectious diseases to childhood deaths out of the newborn period. Many newborn deaths are also due to infectious diseases, including pregnancy malaria. But malaria alone, the 7% of childhood deaths after the newborn period is really all due to infections with a single species of a single organism, as opposed to the diverse organisms that cause pneumonia, diarrhea, et cetera. So it's really an incredible burden. And we're going backwards in terms of malaria control. So the COVID-19 pandemic was really a wreck on public health services around the world. And as a result, in part, due to COVID-19 and other factors, the deaths and case rates of malaria have gone up in the last several years, really, unfortunately. So malaria is an infectious disease, as I mentioned, and it's a parasitic disease. It's caused by infection with a unicellular protozoan. There are 5 species in the genus Plasmodium that cause disease in humans, but almost all the severe malaria and the deaths due to malaria are due to infections, as I mentioned, with a single species, which is Plasmodium falciparum, shown in the image there. So this is a parasite that lives inside human red blood cells, and you can see it infecting red blood cells in culture there. It really doesn't matter what the life cycle is for the purposes of this talk too much, but this is a disease that's transmitted by mosquitoes and then cycles in and out of red blood cells, as I mentioned. And relevant to this is really that this cycle inside red blood cells is what causes the clinical malaria symptoms. So this is the cycle of the disease that we need to detect in order to diagnose this infection. And this is also, of course, what causes the symptoms and the death. We've recently, we see actually a lot of imported malaria from travelers at our institution at the Children's Hospital of Philadelphia, and we recently did a epidemiologic study of these imported malaria cases, and this was work led by Brian Roper and Saish Sundar Arman in my lab. And I bring this up really because it highlights one of the biggest challenges in malaria is really diagnosing it. So in our own cohort, in what I think is probably the best, if not one of the best children's hospitals in our country, if not the world, almost 1/4 of the children who ended up diagnosed with malaria had a previous healthcare encounter where their malaria diagnosis was missed, which is, potentially catastrophic. And many of these were in fact, our own emergency room. And so it really hides one of the biggest challenges in malaria control, which is that everyone who has suspected malaria needs to be tested. Obviously, then anyone who's positive needs to be treated, and we need to track those in order to do malaria control. And it's very analogous to what we all recall needing to do during COVID-19. But this challenge about everyone with suspected malaria being tested is a particular challenge, especially in low resource settings. So, malaria, the most common symptom is fever. And that means if you are a child under the age of 5 living in a malaria endemic area, every single time you get a fever, you need to be tested for malaria. And so to put that in perspective, your average 2-year-old, if any of you are parents, you will know this, your average 2-year-old has about 8 to 9 febrile illnesses a year, right? 8 or 9 times they're going to have fever due to something. That means 8 or 9 times that child needs to be taken to a healthcare provider and tested for malaria. This is an incredible burden on the healthcare system, and it's also an incredible logistical challenge. And so right now, the only diagnostic tests we have for malaria require a blood test. Obviously, this is not popular with your average 2-year-old. And historically, malaria has been diagnosed just by looking at the blood under the microscope. You get the blood sample, you smear it out, and you look at it under the microscope. And really revolutionary have been the development and implementation of these rapid diagnostic tests. So they, just like a COVID test or a malaria, a pregnancy test, 1 line is negative, 2 lines is positive, really simple for low resource settings, but you still need a blood smear. And unfortunately, these rapid diagnostic tests are under threat. So as many of you will recall from Jurassic Park, life finds a way. And as soon as these rapid diagnostic tests got implemented and widely distributed around the world, the parasite stopped making the protein that they detect. And so in multiple parts of the world, parasites have evolved that lack this protein, HRP2, that's detected by these rapid diagnostic tests. In parts of the world like Ethiopia and Eritrea, up to 80% of the parasites can no longer be detected by rapid diagnostic tests. So there's an incredibly urgent need for new malaria diagnostics to really implement for both immediate healthcare needs and also for malaria control worldwide. So this is just again, a sort of schematic of that problem, which is, of course, if you have the antigen, you can be detected. And if you're missing that antigen, the test is false negative. So my lab has been long interested in volatile organic compounds produced by the malaria parasite. We were interested in this originally from some preliminary data suggesting the parasite was actually making compounds that could be detected by the malaria transmitting mosquitoes. And so this is just some now relatively old data from my lab where we looked at cultured malaria parasites and looked at their headspace gas. And what we found in the headspace gas of cultured Plasmodium falciparum is that they make actually sort of a rich abundance of terpene biomarkers. And so these are the same sort of plant-like terpenes that we've described before as being microbial origin or dietary origin like limonene and cymene, but they're not present generally in uninfected red blood cells and appear to actually be coming from parasite metabolism. And so when we first did this study, of course, everyone kept asking me, well, okay, they do this in culture, but is this actually happening during real human infections? And so many years ago, we started by asking this question: do children with malaria smell different? Are there different VOCs present in the breath of children with falciparum malaria? And so we partnered with 2 of my colleagues, Indy Trehan, who was at that time based in Lilongwe, Malawi. Chad Schaber was the graduate student in my lab who did this work. And we did all of this work in the Kamuzu Center in the emergency room. And we were really looking to identify breath biomarkers that would distinguish children with malaria from children who had other conditions that were causing fever. So these were children who were being seen in the emergency room and being evaluated for malaria. So we weren't looking, for instance, for biomarkers of fever. And to summarize some work that's now published, we were able to utilize this breath collection system. This is just a video of how we do this in the field, where we have children exhale into a bag. We collect generally about 1 liter of or more of exhaled breath from a child and then pump that over thermal desorption columns, which are then shipped to the US for GCMS. And using that initial analysis, we were able to identify 6 breath biomarkers that distinguished children who had malaria from children who had fever from other causes. And if we used a cumulative abundance metric of these discriminatory VOCs, we were able to diagnose malaria with reasonably high accuracy. Of course, this was a pilot study in a small cohort and really required validation. And I'm pleased to say that we have now completed that validation in a completely independent cohort of children. And so this work has been done in collaboration with Carl Seidel, based at Blantyre, Malawi. So also Malawi, but a different institution. And in this case, we enrolled febrile children. So again, children coming to the emergency room with fever under evaluation for malaria. We also were able to enroll a lot of healthy siblings that were sort of along for the ride, as it were, in the emergency room. And that gave us a healthy child comparison cohort as well. Over 100 children total in this study. And we evaluated children by rapid diagnostic as I showed you before, but we also did qPCR on all of the children to sort of definitively assign whether they were in the group of malaria positive or negative. And in the children who were RDT positive, we had them return on day 3, so we could actually repeat both blood smears and breath sampling so that we could see how breath biomarkers actually respond to antimalarial treatment. So this is just a schematic of the overall sort of approach and assignment. We ended up with 29 healthy children, 30 children who were uninfected and febrile, 56 children who were Plasmodium falciparum infected. Once we did qPCR to confirm our group assignation, we had 28 healthy, 26 uninfected febrile, and 61 who were Plasmodium falciparum infected, as there were a couple patients who were thought to not have malaria whose qPCR was positive. And then we had pretty good compliance with returning to clinic. So we had 48 children who returned 3 days after treatment with the standard antimalarial artemisinin to get repeat VOCs. An important control that we use is a 24-hour dietary recall in children who are in our pediatric studies, because as has already been shown, diet makes a pretty big difference to breath biomarker identity. And fortunately, our groups were broadly similar. This is a part of the world where diets are generally monotonous. And so across our study groups, the diets were fortunately similar, which really improves our yield for biomarker discovery. And we also had a nice range of parasite densities. So a lingering question was really, do the biomarkers that we identify only correspond to patients who have high parasite densities? Can we also see malaria distinct volatiles in the breath of children who have low density parasite infections as well? And so we had plenty of subjects in each of our groups in order to be able to answer that question. So this is just a principal components analysis, really demonstrating that we confirmed again in this larger study that malaria has a very distinct breath VOC signature. You can see how distinct the children are. In this case, each point is a child. You can see how distinct the breath is of children who are healthy and don't have fever. So you could even group the malaria positive and negative febrile children together. You can clearly see the difference between healthy and sick, which is what really highlights how important it is to have this febrile malaria negative control group. Otherwise, you would really have interpreted all of this as being due to malaria. And you can see how tightly our malaria positive children cluster. So obviously the malaria negative children are in have a wide variety of different infectious and non-infectious inflammatory conditions, whereas the children who have malaria generally cluster much more tightly. And so that really, again, gives us confidence that what we're seeing is a VOC signature that is really quite specific to malaria. We used a number of different methods, including a Barada feature selection. This is a machine learning method that allowed us to really, in an unbiased manner, identify which were the important or unimportant features present in the breath that distinguished our different groups. In this case, this is just showing one of these where we've got low, medium parasitemia of malaria versus febrile controls. And you can see we can identify some specific features in this case. And you can see using the Barada, again, a sort of cumulative abundance metric that clearly distinguishes symptomatic patients with malaria from those who are febrile but don't have malaria. Oops. Here we go. And we have identified some promising breath biomarkers for malaria from this. Nonanol was a compound that arose in our original pilot study, and we were able to again confirm that this is a compound that is specifically enriched in the breath, or specifically distinct in the breath of children with malaria. In this case, you can see the symptomatic malaria negative children versus the symptomatic children with malaria versus the febrile malaria negative children, and you can see the clear differences in this compound between those two groups. As many people have noted already in this conference, acetone is a canonical breath biomarker, breath volatile. In this case, we think it's probably reflective of relative low dietary intake in the setting of illness. And so you can see acetone is enriched in the breath of children when they come in with initial malaria, and after treatment, it responds really nicely to antimalarial treatment, as you would certainly expect. But our overall cohort has given us some ability to identify breath biomarkers that actually respond to antimalarial treatment, which is particularly nice when you think about using this as a screening test. Our study has led to a lot of remaining questions, which we hope to answer in the coming years. One of these is we've really focused on pediatric malaria, in part because this is the group that we feel like for acute symptomatic malaria, really diagnostic test will be most useful. They're the population that's going to need to be screened most readily. But another potential use case for a malaria diagnostic is really to detect asymptomatic infections. So when you think about controlling this parasite and controlling the spread, it turns out that many people who live in malaria endemic areas don't actually have any symptoms. As we noted in our study, there are children who are literally running around playing football in the field and yet have parasites in their bloodstream. And so those children who are asymptomatic but have parasites are at risk for transmitting to other individuals who then might get very sick. And so to really control malaria spread, we need to be able to identify this asymptomatic reservoir of infections, which in some parts of the world can be as much as 80% of the people who are actually infected. And so our studies to date have really not addressed this question because our N has not been high enough of whether or not these breath biomarkers are actually present during asymptomatic infections. And so fortunately, we've recently obtained funding from the Bill and Melinda Gates Foundation to answer just this question of whether or not we can non-invasively diagnose asymptomatic falciparum infection. And fortunately, we have the means here to actually do both biomarker discovery and validation in our single study. So we're going to be enrolling simultaneously in 2 different countries, Malawi and Zambia. And ages 4 and up, so we'll be collecting both children and adults, doing initial screening via rapid diagnostic tests with individuals who are positive or negative, hoping to obtain sort of a balanced set to obtain breath collection from those individuals, both using our standard breath collection device and the Alstone Receiva device to compare those 2, particularly in children. We'll also be collecting skin biopsies, again, sort of hoping to identify asymptomatic other biomarkers that are non-invasive that will detect asymptomatic falciparum infection. And then we'll, I guess, as I mentioned, both do targeted validation of the biomarkers we've already identified, and also unbiased discovery in both cohorts with validation across cohorts, so that hopefully in one study we can really get to answer this question definitively of whether or not you can really have a malaria breathalyzer that could be used for population-based screening and control. So that's really all I wanted to share with you today. Obviously, I want to thank both our funding sources and the many folks in my lab who have their own TikTok. I do not have anything to do with this TikTok, but this is our lab's TikTok. And you can see many of the members of my lab here. Particularly want to highlight Amalia Berna, who is an analytical chemist that I've worked with for many years, and Yang, who also works on our GCMS and has been incredibly important to our studies. So thanks again, and happy to take some questions.
Wonderful. Thank you so much, Audrey. That was fantastic. Really appreciate it. So quick reminder, everyone, put the questions in the Q&A. I was hoping to kick it off with two quick questions myself. So for some of the identified VOCs, I was wondering if you might be able to elaborate a little bit on the potential mechanisms. Things like nonanol probably relates to oxidative stress or inflammation, but maybe the terpenes as well. I was wondering if you could kind of comment on why some of these things may be changed in infection.
Yeah, it's an important question, because I think understanding the underlying molecular mechanisms of how these breath biomarkers change is really going to help us answer the question of specificity of whether or not you expect, for instance, nonanol to only ever be specific to malaria versus other pediatric infectious or inflammatory diseases. And really, we don't have a good answer for most of them because we haven't yet tracked all the carbons. Hopefully, animal models will help us here. I do think the terpenes come at least in part from the parasite itself. So our work in vitro shows that the parasite can absolutely make its own terpenes. And if you block parasite metabolism, you block terpene production. And these terpenes have been identified both in our studies in pediatric malaria, but also in murine malaria. So I think that's real. And so I think that's at least in part one mechanism, but I think there are many other potential mechanisms. And I think that's a really important ongoing question.
And the terpenes are really interesting. I saw some of James Logan's work where it's potential that the VOC is attracting the mosquito. Do you think that is likely to be the case, or a potential reason for why these are being produced?
I think that is a compelling question. So there have been multiple studies in humans. There's been studies in mouse malaria. There's even been a study in bird malaria, avian malaria, that shows that mosquitoes are more likely to both bite and have prolonged biting of individuals who are malaria infected as opposed to those who are uninfected. It's a lot going on there, because when you're infected, especially when you have a fever, your CO2 is higher, your temperature is higher. All of those are mosquito attractants as well. And so it's a really compelling hypothesis, but it's not yet, I think, been fully proven.
Fantastic. On the classifier from the first study, so you had the cumulative abundance and then a threshold for the cumulative abundance of those individual VOCs. Were they equally weighted, or was there weighting given to different VOCs within that classifier?
In that classifier, we equally weighted. It was really a proof of concept. Could you use VOCs to classify? It looks like, at least based on that study, you could. And so that really was the preliminary data that I think supported a larger study.
Fantastic, fantastic. And then in the follow-on study where you showed the PCA, was it all the same compounds that were studied in that follow-on work, or were there new VOCs found from untargeted discovery as well?
Absolutely, all of that was from untargeted discovery. So we did look at targeted discovery, looking at the previously identified VOCs, but all of that initial analysis in the PCA was based on untargeted analysis.
Fantastic. Next question here is, do you think other parasitic infections might cause similar changes to breath volatiles?
Yeah, I think that's an important question. I think it really gets at this question of how you validate these kinds of studies writ large, because there's no way to, for instance, have the perfect cohort that includes malaria and leishmaniasis and Chagas disease and right. And so, because you could just never do that study clinically. So for us, it's really important that our control groups be representative of the control group in the population that we would ultimately want to test. And so that, for instance, is why we wanted to compare children with acute febrile malaria to children who were thought to potentially have acute febrile malaria, because that's the group we're really wanting to look at. So I think it really gets at what is the biological origin of breath volatiles. If, for instance, there may be a common immune response to different parasitic infections, protozoan parasites in particular, that is a common outcome of the same breath volatiles, but we really need to understand that in more detail.
Yeah, the next question I think ties into that in the previous point we had. Nonanol is also found in asthmatics' breath. Do you think it's related to any inflammatory pulmonary process?
Well could be the case. And since non-anol was actually reduced in our children with malaria, maybe it's because the children who are febrile from other things have acute respiratory viral illnesses and therefore increased lung inflammation, right? Would be one possibility.
Yeah. Next question here is, could you please comment on the safety issues in handling of malaria positive samples, particularly sending and handling of the TD tubes?
Oh, that's no problem for malaria. It was actually much a greater challenge for our SARS-CoV-2 work, which I didn't talk about today. So malaria is a blood-borne pathogen, and even the blood sample itself is relatively safe because this is an infection that's spread by mosquitoes. So essentially, unless you're either going to inject the blood into yourself or allow some mosquitoes to bite and wait 2 weeks and then bite somebody else, we're pretty safe from getting malaria. So handling the blood samples themselves, the folks that are doing it in country who are actually collecting blood samples and analyzing those for qPCR, they are at risk for other bloodborne pathogens, but they're not at risk for malaria.
Fantastic, thank you. Next question here is, maybe I missed the information, but is there any correlation with severity of disease?
It's a great question. It's a much harder question to answer because the more severe you get, the harder it is to get those breath samples from children. So as the predominant presentation of severe malaria in children is coma. And so obviously a child in coma is not capable of giving voluntarily a breath sample. And so children with altered mental status or coma are really not a population we can collect from. So we're actually now have strategies to collect breath from infants and other individuals who can't cooperate voluntarily with sampling. And so I think that is a question. But for the purposes of our study to date, we've exclusively looked at these febrile kids, simple febrile, uncomplicated malaria.
Fantastic. I had one quick question. So do you think there's any change to the liver or liver function during infection? And the reason I'm asking that is obviously a lot of VOCs you would expect to change if there's any alterations to the liver.
That's going to be variable, and that's also going to be variable among the malaria negative group. So, many acute viral infections in children will have some transient hepatitis associated with them. It's actually pretty common even with things like influenza, and so really hard to know how that might be distributed among our cases and controls in this study.
Got it. Thank you. Next question here is, could you tell us a bit more about how you plan to sample skin VOCs?
We have been working on this because we do find this a challenging thing to think about doing in a very resource limited setting. Right now, our plan, and we're not definitive on this, is to actually do skin sampling using salt. So actually rubbing salt and then collecting the salt as a source of skin VOCs. It's very easy. It's probably not the highest yield source, but it has a lot of ease and safety, which not all the other potential sampling methods do.
Very cool. I saw a really interesting device just last week in the US, which was a series of micro needles that you attach. It's a skin patch essentially. So they're thinking about it for a way to put drugs into the body, but also as a way to sample interstitial fluid, which I thought could be a really interesting way to think about another non-invasive sampling matrix where you could do headspace VOC type analysis. So
that's interesting.
I'll send it over to you. I was like,
please do.
I was like, I need to learn more about that. So time for one last question here. So malaria generally occurs in low resource populations. Could you please shed an idea on how much the total cost may be for a test once it's fully developed?
Yeah, also a really important question. So the target for the WHO is well less than $1 a test. And so if you think of the kind of sensors or sensor arrays that are under development, I think most folks are targeting that sort of a sensor array, once it's tuned, should be in that sort of $200 to $300 range. Assuming that that's the case and those can be reused, then I think that's very cost effective because you could test over and over and over, obviously, on individuals, and your cost per test will be well sufficiently within the range. I think that initial outlay is still going to be a challenge, but I think if you've got a robust test that really allows you to screen a population, it will still be worthwhile.
Wonderful. And on that note, thank you so much, Audrey, for sharing your wonderful work with us today. I really appreciate you taking the time. And we can't hear the party outside too much, so the playing on Zoom seems to be working. Have a great rest of the day, and thank you again for joining us today.
Thanks so much.